Hi everyone, it's Mr. Sinti, and today let's talk about DNA synthesis or DNA replication. What's interesting is this molecule DNA, the double helix. Here it's sort of looking like a ladder. When DNA replicates and makes two copies of itself, which are completely identical, if you think about it on a molecular level, it's the most essential concept because it's these molecules that are replicating is basically at the most fundamental level responsible for reproduction of all life on the planet. And so let's get into this. As you can see here in the molecule, you'll see, let me highlight some of these areas right here, and this will refresh you about the structure. You see if I circle this right here, this is one nucleotide. This is the phosphate, which is not shown, the sugar deoxyribose in a nitrogenous-based cytosine. One connected to another one, connected to another one, connected to another one. So this is a polynucleotide. We said the DNA is double-stranded, which means that over on this side over here, there's another nucleotide, another one, and another one, and another one. Now, of course, this goes on in this direction, this direction. There's around 3 billion base pairs in human DNA. So if there's two strands, how are they held together? They're held together by hydrogen bonds, and then they coil into the familiar helical structure. So one of the things that we have to sort of contend with when we talk about how the cell replicates its DNA is how is it going to uncoil the helix and get the job done. So there's a couple questions we have always about a process like this. It's like, how does DNA replication occur? And that's important. That'll be the gist of the conversation. But before we get into that, let's talk about like why does DNA need to be replicated in the first place? And we could even talk about where it occurs and when it occurs. Those are relevant questions, and so we should approach that. So let's jump right into this process, if we may. Let's go here, and then let's do this. Full screen. Okay, so DNA synthesis. Let's jump into this. Well, in a eukaryotic cell, that is a cell that possesses a nucleus, DNA, as you may know, is restricted to the nucleus based on size, and so one might imagine that the replication of DNA occurs in the nucleus, which is right here. This is a plant cell. But let me caution you that as you know from many discussions in the past, most living things on the earth are prokaryote, meaning not possessing a membrane-bound nucleus. And that's altogether not a defect. And so if there's no nucleus, if the DNA, like for example in a bacteria, there's one circular big piece of DNA, one chromosome in a bacteria, this simply replicates itself into a second one, like that. And it's got to do that before the cell divides. And so replication in a prokaryote occurs in the cytoplasm and a eukaryote in the nucleus. What am I showing you here? This is a typical animal cell. Here you can see the plasma membrane, here's the cytoplasm, here's the nucleus. Now when I look in the nucleus, I see all sort of like strands in here. And so when I see strands, and this is what scientists saw when they first saw cells under the microscope, all this sort of colored material or chromatin is sort of loose, as opposed to when the cell needs to divide, it undergoes mitosis. And so it takes its loose DNA and coils it up into chromosomes. Let's talk about that for a second, because this is relevant to the question of when does replication occur. So replication occurs at this time. This is a typical cell during interphase. You may recall that interphase is a long period of time. It's the time in which the cell grows, it synthesizes protein, and about halfway through interphase, it takes time to replicate its DNA, and then you have two copies. And the reason you do that is you're anticipating the cell dividing. So if the cell divides, you're going to need two copies of DNA. So DNA replication occurs during interphase. And then let's just talk about this. After interphase, the DNA coils up into chromosomes. You can see it here. The nucleus is starting to break down. You might recognize this cell as prophase, the first phase of mitosis. So chromosomes form, and then they line up in the true equator of the cell in order to organize to make sure that you don't lose any of your chromosomes. And then they get pulled apart during anaphase. It's a great picture of anaphase. and then they arrive on opposite sides of the cell, or telophase or telophase, and then you can see here the cell starting to pinch in. So those are the 4 phases of mitosis, and then you have cytokinesis, and the cell divides into 2. Well, if you recall this diagram, this is part of the cell cycle. This happens in a typical cell. So you can look at this. We were starting right here. We were looking at prophase. So we said the chromosomes form, they line up in the middle, the chromosomes are pulled apart into sister chromatids so that it's an equal distribution to both sides of the cell. The sister chromatids arrive, and so you have a cell like this, and it's just starting to pinch into 2 like that. And so this is the point. If you have your DNA over on this side and your DNA over on this side, then you're good to go. You're good to divide. And so cell division occurs right there at the end of mitosis, or cytokinesis right there. And so then you have 2 daughter cells, and each of those cells will go through interphase all the way around right here. And as you can see here, G1 is growth, and then right here is where I want to emphasize. So about right in the middle of interphase, you get this period called S, or synthesis. This is where DNA replicates. So when does it replicate? When does a cell replicate DNA? During interphase. And so let's, now that we know where it occurs in the nucleus, when it occurs during interphase, let's jump into the details. So this is a bunch of cells in interphase right here. It's the most common phase to be in because it takes the longest. It has the longest duration. So let's talk about now how does replication occur. And so remember I said one of the challenges is that the molecule is a helix. And so you got one strand going like this, the other one wrapping around. So in order to replicate it, you've just got to break the helix. You've got to unwind the helix. And the simple way to do that is break these hydrogen bonds which are holding the 2 strands together. Remember adenine and thymine pair and cytosine and guanine pair. So if you just break those hydrogen bonds, you might have guessed it, there's probably some enzymes involved which are catalysts that speed up chemical reactions. And so the very first thing that we want to consider is that, as it says here, a human cell can copy its DNA and divide it into daughter cells in just a couple of hours. So if it's going to do that, probably means that there's enzymes involved speeding it up, probably means that there's more than one enzyme at a given time doing it. It'd be more efficient. And so many enzymes involved, and there's some complexity to this process, but I thought for this presentation I'd keep it rather basic and just look at the most important ideas. Not to say that the other components, the other enzymes are detailed and trivial, they're not, but for this discussion we'll just keep it loose and we'll just sort of, pun intended, meaning like loose DNA. So the helix needs to be unwound, and so the most important enzyme that does that is an enzyme called DNA helicase. It's a great name. It's easy to remember. It's the enzyme ACE that is unwinding the helix. And so it untwists and separates the molecule by breaking, if I were to add this in my notes, I'd say by breaking the hydrogen bonds between the nitrogenous bases. So you see here what looks like a big bunch of grapes, right here, purple grapes. These are the amino acids of the enzyme helicase. And it basically attaches to a particular area. This is something that we're going to want to talk about later, but it attaches to origins of replication. And so there are specific sequences where it knows where to attach. Do you see how when it pulls it apart it forms like a little fork right here? Obviously DNA continues around up here and up here, so the truth is it actually forms a bubble, a replication bubble. And how do we know this? We actually have pictures. This is a transmission electron micrograph showing this. And so you can see here there's one double helix, and then you can see how it's broken up into 2 right here. So this is one strand and this is the other strand, and this is a little bubble forming, and then there's another bubble forming. And so where would the enzyme helicase be? The enzyme helicase would be right here unwinding it, and it would also be over here unwinding it. So it unwinds it here and unwinds it here. Pretty interesting. It's like early on in our understanding of replication, we sort of just figure things, and we sort of like create models, and we sort of can guess. how the process is working. And as our technology increases, we're now able to see these things, and it sort of confirms earlier hypothesis. So that's kind of neat. So there's areas where the molecule is replicated, and those are origins of replication. And so what happens is, this is what we've been talking about. So this is like that little fork right here. Here's the enzyme helicase. What's it doing? It's moving around, breaking the hydrogen bonds. So now you can see the exposed nitrogen bases here and here. If you put them together, you notice A and T, G and C, C and G, T and A. So these were once together, but now they're broken up. So the idea is you want to copy this strand and copy the bottom strand. If you copy both strands, you'll have two complete copies that are identical to one another. So how do you do this? You basically have to do what's called a polymerization. Let me write that out. Polymerization. So polymer is what we're talking about. Polymerization, this is a polymer. And the reason that it's a polymer is that it's many monomers, which are the nucleotides strung together. So the enzyme involved here, there's going to be many, but I'm only keeping it to two. The first one unwinds the helix, and the second one, which is called DNA polymerase. And it's the enzyme that speeds up the polymerization of new DNA. So it comes in, for example, and it grabs what I would like to call free-floating nucleotides. You're like, well, where are we? We're in the nucleus right here. Let's do this, draw the nucleus. How did these nucleotides get in here? Well, they simply diffused into the nucleus via the nuclear pore. And I'm not showing detail here, but this is like phosphate, sugar, base. Phosphate, sugar, base. Phosphate, sugar, base. And so this enzyme, DNA polymerase, comes around, grabs one of these nucleotides and attaches it. For example, if there's an A there, it would attach a T. And then this would be a C. And then this would be a G. And then it would do this. It would be an A. And this would be C. And this would be T. This would be C right here. Do you notice how, if you look down below, this is exactly what the molecule looked like originally, A with T. So down here you'd have to put an A and then a G. See how this is working? And then a T and then a G. So ultimately you're making two copies that are exactly the same. So the enzyme polymerase, let me just say that again, it adds new nucleotides to make two complete DNA molecules. And so it's responsible for the actual replication. It's the one that catalyzes or speeds up the elongation of the new DNA strand. And so here's kind of a cartoon drawing of it. Here's the enzyme moving along, and it's bringing in free-floating nucleotides. Where do we get these nucleotides? We eat them. They're in our food. And so these nucleotides come in, we eat them, they go into our digestive system, they're absorbed into the blood, and then they diffuse into cells. And so it's interesting, the enzyme works pretty quickly. And so this is a bit of a trivial point, but we never talk about how long things occur or how fast they occur, but the elongation is about 50 a second, so 50 nucleotides every second. It's pretty quick. And so this is, again, something to consider. I like this diagram. It shows the original DNA molecule here in dark blue. So here's the sequence of nitrogen bases. Remember, the sides are phosphate, sugar, phosphate, sugar. So if the molecule opens up like this, and if you remember the enzyme that does that, it's DNA helicase. That's right. So it opens up into two template strands. And then these sort of light blue nucleotides are the new ones. They're coming in from our food. And so the DNA polymerase comes in and attaches it here and here and here and here. Notice how one molecule is the same as the original, and this one over here is the same as the original. So I'm not sure if you were thinking, when you were thinking about replication before we talked about this, if there was one original molecule and then there was a copy of it. Because the fact is, the two new molecules that are created in replication is not like an old one and a new one, but rather an old connected with a new, an old connected with a new. Sort of like a freshman that's paired up with a senior, or if you will, if you remember this line from Yoda in Star Wars, always two there are, a master and an apprentice. So in biology we call this semi-conservative, meaning like a semicircle's half. So it's half old, half new, half old, half new. And so you can see the two new daughter strands. So now there's two complete DNAs. One's half old, one's half new, half old and half new. And both those pieces of DNA are just like the original. Where does this occur, the nucleus? When does it occur? During S phase of interphase. And so I like this picture. It's kind of a cool cartoon. These are the free-floating nucleotides. Here's the helix opening up. The enzymes are not shown. It's fine. And DNA polymerase is adding the new nucleotides. And so here are the two new strands. And so historically, what's interesting is Jim Watson and Francis Crick, in their second paper, which wasn't as popular as the first, where they determined the structure of DNA, but they did write a second paper and posed a hypothesis about how DNA may occur. And it's called the semi-conservative, like we were talking about, replication. Another video coming in the future, we'll talk about, maybe not this year, but we'll talk about the details of this, and it's really cool. There's a scientists, Frank Stahl and Matthew Messelson, that won a Nobel Prize for determining the semi-conservative experimentally nature of DNA replication using radioactive and heavy isotopes of nitrogen. It's kind of cool. So you can see here, here's the doubled helix, one DNA molecule, and then here you're going to have two DNA molecules which are similar. And I want to point something out to you, that if this is the original and then here's a copy and here's a copy, you might ask this question, or not, but I'll pose it. How does this relate to the chromosome that occurs during prophase? You know how we talk about a chromosome as being coiled up DNA like this around histone proteins, and there's one sister chromatid and the other sister chromatid, and it's held together, go a different color, in the center by a center protein called the centromere. And then the whole point of mitosis is to do what, you remember? Is to sort of line these up in the middle and then put one sister this direction and one. That's mitosis. Well, how does this relate? Well, as it turns out, let's go here, blue. As it turns out that this molecule right here, the old new, the one complete molecule, let's coil that up. That's one sister. And then over here, let's coil it up around histone proteins. That's another sister. And then here's your centromere. So you can see the cell does not need a chromosome. It only needs a sister chromatid because this is the complete DNA. That's why during mitosis, one sister chromatid goes that way and that one goes the other way. Kind of explains something. I find that to be, let me just pause for a second, I find that that's a really important point, because it's these kinds of details of how DNA replicate. If you connect it back to something like mitosis, that's when you really understand what's going on, when you make those kinds of connections. So again, one side of the chromosome is identical to the other side. And we mentioned that before, but now you know why. Because this is a new copy and this is the other copy. These are the two daughters. This is an incredible scanning electron micrograph of a chromosome. So this is one new daughter and this is the other new daughter on this side. And so let's jump back to, this is the last slide, and this is showing the prophase where the chromosomes. So what's interesting is it looks like sort of just like black worms right here, but if you were to close up, there would be a lot of these chromosomes inside the nucleus. Isn't that neat, like that? And so let's jump down here. I'm not sure if I wanted to do that. Maybe I did. Let's go up here and let me see if I could do this. So just in summary of what we're talking about, here's the cell like this. Here's the plasma membrane. Here's the nuclear membrane. I'm drawing it like this because I'm not drawing the double membrane, but you can see the nuclear pores. And then over here is the blood. This is where the nucleotides are diffusing into the cell, not showing the detail of that. And so these are the building blocks of DNA. And each one has a little nitrogen base sticking off like this. And you're like, okay, I'm ready to go. So, whoops, here's the DNA like this. And one of the dilemmas is that it's in a helix, but the enzyme, if you remember DNA helicase, opens it up into the bubble. Then the bases are sticking out like this. And if I had an A, T, C, and G right there, I would simply, the enzyme DNA polymerase would simply come around and grab a T and put it there, and then an A would go there, and then a G would go there, and a C would go there. Likewise, this would be T, A, G, and C. And so then the complementary one would be T, C, A, T, C, and G. Then you'd have these two strands that are identical to one another. So let's try this. Let's try going to the desktop, and then we'll go here. I have this, which I think is kind of interesting. Let me make it a little larger to see it. So this is a little animation of what we've been talking about. Perhaps it's a lot better than my drawing. But here's the DNA double helix. This big green guy looks like DNA helicase because it's unwinding the molecule. These are the free-floating nucleotides that are in the nucleoplasm. They're all cruising around, and so the enzyme DNA polymerase is like, okay, I gotta bring them in. And so this is a polymerization because we're increasing the polymer. And of course, this is happening on one side, and it's happening on the other side. You might notice that they're going in opposite directions. That has to do with the fact it's a bit of a detail. Here's the replication bubble right here. It's a bit of a detail, but it's because the molecule's anti-parallel, meaning that the one strand's going in one direction, the other one's going in the other direction. It's not too much of a detail. We'll talk about it in the future, but not in this particular video. And let's conclude with this. This is an animation of, this looks like endoplasmic reticulum. We're going to go into the nucleus right here. You can see we're going in through this little pore right here, nuclear pore. So what do you expect to see inside? Well, if replication occurs during interphase, I would expect to see lots of strands of DNA, sort of like spaghetti just hanging all over from the ceiling. And so this is what we see. The DNA is loose, it's not condensed into chromosomes, or else replication wouldn't be able to take place. So here's a close-up of the DNA, and if we sort of get even closer still, you can see how this green area is phosphate sugar, phosphate sugar, phosphate sugar. These are the sides of the molecule. But what's most important are the rungs, or steps, because that's the sequence, that's the genetic code. This is how we're different from one another and different from other organisms. So when DNA copies itself, fundamentally this is how cells are similar. They're able to copy their DNA, and that's how mitosis is able to move the chromosomes around. So in order to replicate DNA, what we're going to need to do is uncoil the helix. So we have to break these hydrogen bonds. And so what's interesting is you can apply heat. We're going to find out in the lab that heat would be able to break this bond, but in the cell you don't want to apply that kind of heat. So here's the enzyme in purple here called DNA helicase, and it's coming along. It's kind of weird how stuff like this is happening in the cell right now as you're watching this. But here's the enzyme, and it's breaking those hydrogen bonds, and it exposes, see, the bonds are breaking between the nitrogen bases. So now you have two strands on both sides here, and this forms the fork. And so up here, these unincorporated nitrogen bases are sticking out. And so here in blue is the enzyme DNA polymerase. It comes and it grabs free-floating nucleotides and it attaches it in place. And so basically, if there's an A there, it's going to bring in a G. If this is a C, it's going to bring in a G. And so it goes along and it elongates or polymerizes the DNA molecule. One could say this is, as you may know by now, I'm prone to hyperbole, but this DNA polymerase is a really critical, really crucial enzyme. It's fundamentally responsible for reproduction, which is pretty important. In addition to just polymerizing DNA and making new DNA, it can even proofread. It can determine, like maybe it makes a mistake, and it's like, hey, the molecule doesn't fit, and then it brings in the correct nucleotide. Every once in a while, just a little foreshadow, every once in a while a molecule like this makes a mistake. And so the sequence is slightly different. And you're like, tragedy, a mistake. Well, a mistake in the DNA sequence or a change is called a mutation. And I know the word mutation sort of has a deleterious suggestion, meaning that it's harmful, but you can only improve if you make a change. Now, sometimes a change is bad, sometimes a change is good, but you can only improve if you make a change. And so this is a very critical discussion in biology, and I'm just going to sort of finish with this, is when DNA replication occurs, and we like to think that it's perfect, but when mistakes get made, in other words, like you make a change to your recipe, it's the only way in which things can improve in the future. So mutations often occur during DNA replication, and therefore there's variation in the future. And variation in the future is something that evolution can work on in order to make changes. So I hope you enjoyed this conversation about DNA replication, DNA synthesis. Thanks for watching.